This is our cell membrane structure, permeability, and transport animation project. This is a phospholipid. The head, which consists of a phosphate group, is both polar and hydrophilic. The hydrophobic tails consist of fatty acids, which are made of hydrocarbons. In an aqueous environment like water, phospholipids arrange themselves into a bilayer. The hydrophilic heads face the external environment, while the hydrophobic tails face the inside of the cell. There are two different types of proteins that are incorporated in the plasma membrane. The first is an integral protein. Interactions between side chains and fatty acids anchor the protein to the membrane. The peripheral proteins are indirectly attached to the phospholipid heads or integral proteins. There are many different characteristics and functions of membrane proteins. Transport proteins move chemicals into and out of cells. Proteins built into the membrane could be an enzyme with its active site exposed to substances in an adjacent solution. Membrane protein may have a binding site that fits the shape of a chemical messenger, allowing the external messenger to relay the message to the inside of the cell. Proteins can sponsor cell-to-cell recognition when they serve as ID tags that are recognized by membrane proteins of other cells. Membrane proteins of adjacent cells may hook together to form various junctions. Lastly, some proteins' attachment to cytoskeleton and extracellular matrix allows them to coordinate extracellular and intracellular changes. The plasma membrane is a fluid mosaic because of its molecular orientation, separating the cell's interior from the outside environment. Glycolipids stabilize the cell membrane surface by forming hydrogen bonds with water, and glycoproteins act as cell surface receptors. Steroids also stabilize the cell membrane, and embedded proteins allow for selective transport of molecules. The membrane's hydrophobic interior prevents certain ions and molecules from directly passing through the membrane. Type 1 ions do not mix well with the bilayer because of their high polarity. Type 2 molecules are polar and large, so they can only pass if they have carbon-based functional groups. Type 3 molecules, despite being polar, can still pass through due to their small size. Finally, since like dissolves like, the nonpolar small type 4 molecules can pass with ease. Embedded proteins are proteins that are fixed onto the lipid bilayer. These proteins allow for the passage of information, waste, nutrients, and water. There are two different types of transport proteins, channel proteins and carrier proteins. Channel proteins have a hydrophilic tunnel that allows polar molecules and ions to pass through the membrane. Carrier proteins carry solids from one side of the biological membrane to the other side. Although water is highly polar, it is able to cross the plasma membrane due to aquaporins, channel membrane proteins that transfer water through the membrane's phospholipid bilayer. The cell wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. Within the wall, specialized cells hold the plant up against the force of gravity. Cell walls are also perforated with plasmodesmata, which allow water and small solutes to pass freely from cell to cell. There are different types of passive transport. Facilitated diffusion occurs when polar molecules and ions diffuse with the help of transport proteins. Diffusion is when substances move from high concentration to low concentration without assistance. Osmosis is the movement of a solvent through a membrane to a more concentrated solution. Active transport requires energy because substances move against the concentration gradient. The sodium-potassium pump works by exchanging a sodium ion for a potassium ion across the plasma membrane in animal cells. ATPase is an enzyme used to catalyze the ATP synthesis reaction, and the ATP produced is used to transfer substances against the concentration gradient. While both endocytosis and exocytosis require ATP, in exocytosis the cell secretes molecules by fusing vesicles with the plasma membrane. In endocytosis, new vesicles are formed as the cell takes in new molecules.